Welcome back to our second part of Chapter 11. So today we're going to talk about evolutionary trees and how they're going to help us conceptualize and categorize biodiversity. So with this, Charles Darwin proposed and documented the species could in fact change and give rise to new species. So with Darwin, the classification of species acquired a new goal and more important functions. The origin of species that Darwin wrote, he said, our classification will come to be, as far as they can be made, genealogies. And so that was a quote from his book. So that's Darwin proposing that the classifications of organisms would resemble family trees that link parents and offspring over long periods of time. So with these words, Darwin was first talking about the link or classification of evolution through time, so linking them through common ancestors. So with this, we're going to talk about the somatics and phylogeny. Somatics is going to be names and arranges species in a manner that indicated a common ancestor that they may share, or the points at which they diverged from each other. And when we talk about phylogeny, we're going to talk about evolutionary history of organisms. So with Linnaean classification involving placing organisms within groups as a function of their apparent similarity with each other, the modern incarnation of Darwin's vision of classification is going to be called systematics. And that's going to be more of a broader goal or reconstruction of phylogeny, or the evolutionary history of organisms. That is, through systematics of all species, even extinct species are going to be named and arranged in a manner that indicates the common ancestors that they share and the points at which they start to diverge and change over. This that we see here is going to be like an evolutionary tree or a phylogenetic tree. Phylogenetic trees are going to be tools that use a root and a series of branches and branch points to trace the evolutionary history of different organisms as they descend from a common ancestor and to illustrate the relationship to these other groups. At the beginning of life on Earth, there was a first living organism, one that could replicate itself and it had metabolism. Then a speciation event occurred. After this event, the population first living organisms split into independent evolutionary lineages. So the phylogenetic tree had its first branch, and then there was biodiversity. It's going to be important to remember that phylogenies are going to be hypotheses, and like any hypothesis, they're going to be subject to revision and modification. Over hundreds of millions of years since then, speciation events continue to occur. Today, the tree branches with millions or possibly tens of millions of tips that represent all different species that we find on Earth today. And it doesn't stop here. Even today, speciation continues to add new branches all of the time. So we mentioned biodiversity. Remember, biodiversity is going to be a variation and total number of species living in a different ecosystem. So also remember that the classification of biodiversity started with Aristotle back in 350 BCE, and we've continued to upgrade and change it throughout time. So with these evolutionary trees, they're going to show ancestor descent and relationships. So many human traditions have a sacred tree of life. The Bodhi tree that the Buddha sat beneath for 7 years to achieve an ultimate truth is the tree of enlightenment. The apple tree in the Garden of Eden was the tree of knowledge. So the history and relationships between all organisms that constitute life on Earth is another tree of life. The trunk and the branches of the evolutionary tree are going to represent ancestor-descendant relationships that link organisms with all life that has ever existed on Earth. So the evolutionary tree of life can be thought of as one giant tree, but also as a practical matter, biologists often study only practical branches or particular branches of this. So these branches can be illustrated as the big tree or little trees, and they can be expanded to include whatever organism the biologist is studying. So the tree might, for example, include all animals, or it might just include things like rodents. So evolutionary trees can tell us many things, but one thing that they don't tell us is which group is most primitive and which one's more advanced. The property of phylogenetic trees can serve to undermine some of the most sacred beliefs, such as one that humans are going to be the pinnacle of evolution. Many trees can be drawn to support that idea, but Notice that if you rotate the tree around one of the nodes, that you can get a number of different trees. So with this, some questions you may ask: are humans going to be more advanced evolutionarily than cockroaches? Well, think about that. Can you live 7 days without your head? Probably not, but cockroaches can. Are you able to survive extreme conditions? For example, if we had a extreme weather event, or if we had some type of nuclear event, chances are the cockroaches might still be living. So would that make them more advanced than us, or are we still more advanced? Can bacteria be considered a lower organism because it's, let's say, single cellular or unicellular, or because we don't see a brain in it? It doesn't have a nucleus. It just simply has DNA, and it has some ribosomes floating throughout. So would you consider that to be a lower organism? Or would you look at it as they've been around since the beginning of time almost? They were some of the first organisms to develop. So those are some of the things that you can think about when considering, are things evolutionarily higher or lower? So what trees are going to tell us is which groups are more closely related to which other groups. One of the most interesting revolutionary revelations of this tree thinking is that despite the appearances, fungi such as mushrooms and yeast and moles are actually going to be more closely related to animals than they are to plants. And so you see them growing out in the wild, you see them in nature a lot. You would think that these things are growing with plants. You would think that they would be more closely related to plants, but they are not. Biologists are going to use the term monophyletic to describe a group in which all the individuals are going to be more closely related to each other than to any other individuals outside of that group. So monophyletic groups are determined by looking at the nodes of the tree. So we consider them to be members of a single, what we call a clade. So a clade's a group of organisms that share one or more symmorphous derived from a common ancestor. And so these members of the single clade include the most recent common ancestors and its descendants. For example, looking here, birds and crocodiles taken together compose a monophyletic group because they share more recent common ancestors, which is designated that you can see by node A, than either group shares with lizards or mammals. Lizards and crocodiles taken together do not compose a monophyletic group because their common ancestors at node B is also going to be shared with the birds. But birds and crocodiles and lizards all taken together do compose a monophyletic group by virtue of all trees sharing a common ancestor at node B. So similarly, birds and crocodiles and lizards and mammals also will compose a monophyletic group when you're looking at larger groups. But here, if you're looking at node A, birds and crocodiles will be the more closely related. Reading an evolutionary tree is going to reveal which groups are going to be more closely related and approximately how long ago that they shared a common ancestor. But how exactly are evolutionary trees going to be constructed? They might hypothesize historical events that happened. In this case, the cat-dog split that happened about 60 million years ago, and that what constructed it to happen in the first place. So that's a good question that we need to look at. That's hard for trees to tell us. Until recently, these trees were assembled by looking carefully at numerous physical features of species and generating tables that compared these features across the species. So basically, you look at clues in body structure. That's going to be a simple example on a table. With only 3 features, this table shows a clear split between the characteristics of a lion and a hyena on one hand, and a wolf and a bear on the other hand. So for most of the 20th century, biologists classifying organisms would often use 50 or more characteristics to generate a tree. Beginning in the 1980s, biologists began using DNA rather than physical traits to generate evolutionary trees. So the rationale for this approach is that organisms inherit DNA from their ancestors, and as species diverge, their DNA sequences also will start to diverge, becoming increasingly different. As more time passes following the splitting of one species into 2, the differences in their DNA sequence will become greater. By comparing how similar the DNA sequences are between 2 groups, it's possible to estimate how long it's been since they shared a common ancestor. So here you're looking at what we call a DNA fingerprint. Now with this DNA fingerprint, they're able to run different species DNA. When they run this DNA, then they're able to look at the differences and the similarities between the two, or between the four that we see here. And so looking at these, we can tell that species 1 and species 2 are only going to differ by 2 base pairs in these areas. And then we look at species 3 and species 4. They're going to be closely related because they're going to differ at 5 different sections. Now, when you compare the groups, you can see that there's going to be a lot more between 2 and 3 that are going to differ than the monophyletic groups that we just looked at. So that's going to lead us to a common misconception. Similar structures are not always going to reveal a common ancestor. For example, whales and sharks have fins. Bats and many insects have wings. So do bats have wings because they're most closely related to insects and inherited their wings from a common winged ancestor? Or did insects and bats kind of evolve together and start to develop wings at the same time but independently? So as evolutionary tree is seen in what we just looked at back in figure 1019, that's going to make it clear. Insects' wings and bat wings are going to evolve independently. And that's going to be an adaptation that's going to arise because of their environment, and it arises over more than one occasion. Phylogenetic trees are going to be based on 2 sources of data. We have what's called morphological data. That's going to be obtained by comparing homologous structures and structures that are going to be anatomically similar in related species due to common descent. For example, vertebrate limbs. Morphological data is going to be an analogous structure, and that's superficial and similar structures in different organisms that occupy the same habitat that arise from convergent evolution and not common ancestry. So they're not going to be useful in determining evolutionary relationships. For example, wings of insects and birds and bats. So looking at this, mapping the species characteristics into phylogenetic trees is going to be basic either physical features or DNA sequence. So like we said, prior to the 1980s, when we started using DNA, they actually went back and they were comparing and decoding according to physical features of organisms. What was present, what wasn't present. DNA sequence then came about in the 1980s, and with that, they're going to start to compare these DNA sequences and determine which animals are going to have common ancestors and which animals are going to be more closely related. Let's look at a case that eliminates why the original methods were weaker. So initially, biologists thought that the African golden moles belonged in the same order of insectivores, which included things like shrews, hedgehogs, and other moles. This belief seemed reasonable because these animals have many characteristics in common. They're all small, they have long, narrow snouts, their eyes are going to be tiny, and they live in underground burrows. But biologists thought that this group of characteristics evolved just once, but that every species in the insectivore order possessed all these characteristics because they inherited it from a single common ancestor. But DNA came around in the 1980s, and the DNA evidence revealed that the African golden moles are going to be more closely related to elephants than they are to insectivores, including all of the other mole species. So that kind of blew everything out of the water when they were able to analyze that DNA. This is going to give us what's called convergent evolution. This occurs when populations of organisms live in similar environments and so experience similar selective forces. So why do they look similar to these insectivores? Because of that phenomenon called convergence evolution. So here we have what are known as analogous traits. These are going to be characterized, things such as bat wings and insect wings, that are the same because they're produced by convergent evolution and not because they're descended from a common ancestor that they share. So lots of times you wonder, what's the difference between the two, analogous or homologous traits? Well, analogous is going to be your traits that feature that are produced by convergent evolution. Homologous are going to be inherited from a common ancestor. So features that you inherit from common ancestors, always think of common, same, homo means of the same type. So all mammals have hair because they inherited that trait from a common ancestor. Similarly, among all reptiles, they have a mouth as a homologous trait because all reptiles share a common ancestor who possessed that trait and gave that to them, so they inherited it. Now, with those analogous features, they're problematic when constructing evolutionary trees because they're a result of a natural selection rather than a common ancestor. So, as a consequence, these traits should not be considered a sign of relatedness between two organisms that have them, but they reflect a sign of things that happened throughout time that caused them to have to change. So that leads you to the question, how would you know whether a trait's homologous or analogous? Well, thanks to the use of DNA, starting, like we said, in the 1980s, we're able to go through the DNA analysis and look at those sequences and see which ones are similar and which ones are not. So if they're not similar, we know it's convergent evolution. If they are similar, we know that it is from a common ancestor. So those are going to be homologous traits. Now we're heading into what we call macroevolution. Macro, of course, means big, so macroevolution is going to be above the species level. So when water runs over rocks, it's going to wear them away. This process is simple and slow, yet it's going to be powerful enough to create things like the Grand Canyon. To be sure, water running over rocks does not always make the Grand Canyon. Nonetheless, no additional physical processes are going to be necessary. The process of evolution has a lot in common with a stream of water running over rocks in the short term. It's going to produce small changes in a population. Yet, the accumulation of these small changes is going to lead to a large change over time. The production of 200-ton dinosaurs from rabbit-sized reptile ancestors, the diversification of flowering plants from a single species into more than 200,000 species, the emergence of animals that live on land. These large-scale examples, which are going to be the products of evolutionary change involving the origins entirely new groups of organisms, are going to be referred to as macroevolution. So these examples can be contrasted with the increase in milk production in cows seen during the first half of the 20th century, or the gradual change in the average beak size of birds depending on the patterns of rainfall phenomenon that's going to involve changes in allele frequencies in a population that's referred to as microevolution. They might seem like two very different processes, but they're not. Evolution, whether it's going to be micro, which is small, or macro, which is large level, is going to be one thing only. It's going to be a change in allele frequencies over time. So that's one thing that you need to keep in mind. These changes over time, or the pace of evolution over time, is not going to be constant. Sometimes it speeds up, sometimes it goes slower. So the traditional model of how evolutionary change occurs was that in previous sections to describe microevolution is going to say that populations change slowly but surely, gradually accumulating sufficient genetic differences for speciation, hence the phrase evolution by creeps. Spurred on by findings from the fossil record that don't always support this view, however, researchers have come to believe that evolution may commonly occur in brief periods of rapid evolution change right after speciation. And that's going to be followed by long periods with relatively little change or no change at all. And so that's referred to sometimes as the evolution by jerks. This newer view on the pace of evolution in which rapid periods of evolutionary change are going to be punctuated by long periods of little change is going to be called punctuated equilibrium. So in nature, there are going to be examples of both gradual change and the irregular pattern of punctuated equilibrium. It's not a situation in which one version is going to be right and one version's wrong. It's just how it seems to happen. So to recap with that gradual change or gradualism, it's basically going to be a process of speciation that occurs when genetic differences accumulate gradually over a series of incremental steps. And then that punctuated equilibrium is going to be a form of speciation that consists of long periods of stasis that's going to be then interrupted by a rapid evolutionary change. So a lot of change really fast. In a brief period of time, a smaller number of species diversified into a much larger number of species, able to live in a wide diversity of habitats. That's called adaptive radiation. Such a large and rapid diversification has occurred many times throughout history. So adaptive radiation is going to give you rapid diversification of an ancestral species into a number of phenotypically unique species that are adapted to a specific environment. And so this could happen with different criteria. We could have the mass extinction events that you see here, colonization events, and evolutionary events. With mass extinction events, it's going to eliminate competition between species and it's going to open new unoccupied habitats. Some examples: the end of the Cretaceous period, we have a mass extinction, and in that extinction of terrestrial dinosaurs is going to lead to large-scale adaptive radiation of mammalian species. Colonization is going to introduce the species to an uninhabited environment with abundant resources and minimal competition. So example of this is rapid diversification of different Hawaiian honeycreeper species that descended from the finch-like ancestors that arrived in Hawaii Islands a long time ago. You can also look at the Galapagos finches that came from Ecuador. With evolutionary innovations, they're going to provide adaptive advantages to species that occupy a particular habitat. So some examples: development of wings and exoskeletons will influence the increased adaptive radiation of insects. Here you see a grasshopper, and so with this, it's going to have an evolutionary feature. It started out with very, very small wings. Those wings were there to help reduce the temperature, so they're there for thermoregulation. The more that they fanned themselves off, the larger that the wings grew. Over time, those wings actually adapted to flight. So they went from being thermoregulators to actually helping them fly. So now they got a completely different species that has developed from something that was there for another purpose. There have been several mass extinctions on Earth. An extinction basically is going to occur when an individual of a species completely dies out. So some things that we no longer have anymore are going to be like the dodo bird, or the Tasmanian wolf, or Tasmanian tiger, as they refer to it. Mass extinctions are going to be rare events that result in loss of a large number of species over a short period of time. They're attributed to volcanic eruptions causing global warming and oceanic anoxia and acidification, global cooling, glaciation events or asteroid strikes. All of these different things can lead to mass extinctions. We have 5 documented mass extinctions in our history. First one was going to be the end of the Ordovician. That's going to be around 450 to 440 million years ago, 450 to 440. Approximately 85% of existing species went extinct. The next one was going to be at the end of the Devonian. That's going to be around 375 million years ago. Then between 70 and 80% of all marine species went extinct. Third one that's going to happen is the end of the Permian. That's around 251 million years ago. That's going to be the one that you see here that says the Great Dying. Approximately 96% of marine species and 70% of terrestrial species went extinct. So that's how it gets its name, the Great Dying. The fourth one is going to be the end of the Triassic period. That's around 201 million years ago. That eliminated about 76% of all marine and terrestrial species. And the last one that occurred was going to be the end of the Cretaceous period. Cretaceous period. That happened around 66 million years ago, and it's going to result in the loss of about 75% of existing marine and terrestrial species, most notably the non-avian dinosaurs. So that was the last one. They actually say we're going through a mass extinction at this point now. A lot of different species, especially in places like the rainforest, are becoming extinct, they say, due to changes in our environment. So different environmental changes are changing the habitats for these animals, and if they can't adapt, they do go extinct. Next, we'll go into what we call background extinctions. With background extinctions, there are going to be common forms of extinctions that occur continually at the rate of 1 species per million species a year. This is attributed to a variety of different factors, including things like global climate change, genetic uniformity, and geographic isolation. Current species vulnerable to background extinctions will be things like polar bears, fritillary butterflies, devil's hope pulpfish, or things like that. They're going to be very dependent upon their environment. So if it changes at all, then they are more likely to go extinct. So with these are going to be your extinctions that occur at low rates during periods other than periods of mass extinction, and they occur mostly as a result of natural selection. So with the mass extinctions, just going back to that, we said they were periods where we had large numbers of species become extinct over relatively short periods of time. And there have been at least 5 that we just discussed. And what happens during each of these is over 50% or more of the animals during that time will go extinct. Background and mass extinctions have different causes. Like we said, with the mass extinction, it's going to be due to extraordinary sudden changes in the environment. Background is going to be extinctions that occur mostly as the result of natural selection. Of the 5 mass extinctions during the past 500 million years, the most recent is also the best understood. 65 million years ago, a massive asteroid smashed into the Caribbean near the Yucatan Peninsula in Mexico. The impact is going to leave a crater that's more than 100 miles wide and probably created an enormous fireball that caused fires worldwide, followed by a cloud of dust and debris that blocked the sunlight from the Earth and disturbed the climate worldwide for months. Now we're going to change gears some and talk about the diversity of life on Earth and organisms and how they're divided into their 3 domains. Remember, those are those 3 groups that we put everything in. So they're the all-encompassing groups. Here you're going to see this is a flower hat jellyfish that you can see. It's going to be a rare species that lives in the oceans off Brazil, Argentina, and southern Japan coasts. So it is a beautiful jellyfish. So all living organisms are going to be divided into one of 3 groups. The classification systems here are going to be a 2-kingdom system, animal and plant first, and then we move to that 5-kingdom system. So Linnaeus' original classification, all organisms were put into either a plant or an animal kingdom. The 2-kingdom system gave way in the 1960s to a 5-kingdom system. At its core, the new system was a division based on distinction between prokaryotic cells, which are going to be those without a nuclei, and eukaryotic cells, which are going to be those that do have a nuclei. The prokaryotic cells are going to be put into one kingdom. That's called the Monera. The only residents in this kingdom were going to be bacteria and single-celled organisms with no nucleus, no organelles, and genetic material that took the form of a circular strand of DNA. The eukaryotes, having a nucleus, are going to be compartmentalized or have compartmentalized organelles, and individual linear pieces of DNA are going to be divided into 4 separate kingdoms, which are then going to be the plant, animals, fungi, and protist. It's going to be a group that includes single-cell eukaryotic organisms like algae. So we'll talk about the protist in more detail coming up in the next chapters. So the classification of organisms is going to take a huge leap forward in the 1970s and 80s when the 5-kingdom system is going to be discarded. So until that point, organisms have been classified primarily based on their appearance. But because the ultimate goal had changed to reconstruct phylogenetic trees to reflect their evolutionary history of Earth's diversity, we're going to have Carl Woese, an American biologist, and his colleagues begin to classify organisms by examining their nucleotide sequence that they carried instead. So now we're going into the DNA. So Woese is going to assume a more similar genetic sequence between 2 species means that they're going to be more closely related. So the only way Woese could compare the evolutionary relatedness of all organisms present on Earth today was by examining one molecule that was found in all living organisms and looking at it at a degree to which different species had changed from one to the next. So he discovered a perfect candidate for this role, a molecule called ribosomal RNA. Ribosomal RNA is going to have the same function in all organisms on Earth, almost certainly because it comes from a common ancestor. Over time, however, its genetic sequence has changed a bit. So tracking these changes makes it possible to reconstruct the process of diversification and changes that take place. The trees Wosey's genetic sequence data generated has some big surprises. First and foremost, the sequences revealed that the biggest decision in the diversity of life on Earth was not between plants and animals. It wasn't even between prokaryotes and eukaryotes. The new tree revealed instead that the diversity among the microbes was dramatically greater than ever imagined, particularly because of the discovery of the completely new group of prokaryotes called archaea. These are going to thrive in some of Earth's most extreme environments, and they're going to differ greatly from bacteria. The tree of life was revised then to show these three primary branches, and they're going to call them domains. So we have bacteria, we have archaea, and we're going to have the eukarya. So bacteria and archaea are going to go in with prokaryotes because they don't have nuclei. The eukarya does. So you would think archaea and bacteria would be more closely related, but the archaea is actually more closely related to the eukarya. So like any type of chart or diagram or creation that you make, it's not going to be perfect. The 3-domain, 6-kingdom approach isn't going to be perfect and is still subject to revision. For example, it seems that single-cell protists are so diverse that they should be split into multiple distinct kingdoms. Also problematic is going to be the horizontal gene transfer in bacteria. Rather than passing genes simply from a parent to offspring, they transfer genetic material directly into another species. This is going to complicate phylogenies based on the sequence of data because it creates situations in which two organisms might have genetic sequence that are similar, but they share common ancestors. And so as a result of that direct transfer, so they weren't common at first, but then they transferred genetic material, which then gave them common ancestors. So that's going to be something that's going to be harder to understand too. So additionally, a fourth group of incredibly diverse and important biological entities comes about, the viruses. It's not even included in the tree of life because they're technically not considered to be living organisms. Viruses can replicate, but can only have metabolic activity by taking over the metabolic process of another organism. The lack of metabolic activity puts viruses just outside the definition of life that we use in the book, but some scientists still view them as living. The most commonly accepted tree of life suggests that after the origin of life, the following sequence of events occurred. First, bacteria arose from the first self-replicating, metabolizing cells. Second, there was a split between bacteria and the line that gave rise to archaea and eukarya. And then third, the fusion of bacterium and archaean-like prokaryotes are going to give rise to eukarya, which then will split from the archaean line. The bacteria domain is going to have a tremendous biological diversity. So why is morning breath so stinky? Well, when you wake up, your mouth's going to contain huge amounts of bacterial waste products. There's going to be several hundred species of bacteria that are going to live in your mouth, mostly residing on your tongue. All are going to be competing for resources that you put there. So those resources are going to be leftover food that you might not have gotten off if you brush your teeth at night. Hopefully you brush your teeth. If not, then they have lots of resources to compete for. Some of these bacteria are going to be aerobic, which means they require oxygen for their metabolism, and others are going to be anaerobic, which means they don't require oxygen. At night, the flow of saliva is going to slow down. Oxygen content in your mouth is going to decrease because usually you keep your mouth closed while you sleep, and the anaerobic bacteria are going to be able to start to overgrow and take over. These bacteria metabolize food bits that were left in your mouth. Plaque on your teeth and gums is going to be left over from the waste products of these bacteria. Dead cells from the lining of your mouth are going to be there, and they also act as food. Breaking down proteins in these molecules to use for their energy source gives them things to live off of. So they don't need oxygen, perfect environment, nice and moist, warm, and they have a good energy source. So as they do, these waste products are going to start to accumulate. So as they eat, of course, they need to make waste. That waste is pushed out of the cell. It accumulates. Because proteins are going to be made from amino acids, many contain smelly chemicals like sulfur. They're going to break down and lead to a bad smell as those waste products. And so that's what's going to give you that bad smell in your mouth or your morning breath. Now moving on, here we see a toothbrush bristle. On a small scale, your mouth is going to reveal some tremendous biological versatility of bacteria. So you're going to have hundreds of species that can live in a tiny area. In a teaspoon of soil, for example, there's going to be more than 1 billion bacteria. They can thrive in a variety of unexpected habitats. They utilize a variety of different food sources, and they can survive and thrive with or without oxygen. Looking around the world, the diversity of bacteria domain is going to be even greater. A survey of the numbers shown the clear dominance of bacteria on our planet. So by a measure, it is their planet, not ours. Because the biomass of bacteria, if they were all able to be collected and dried out and weighed, will exceed that of all plants and animals on Earth. Bacteria live in soil, they live in air, water, Arctic ice, and even volcanic vents. Many can even make their own food, either utilizing light from the sun or harnessing energy from chemicals like ammonia. Bacteria are going to be a monophyletic group. While the various species are going to differ in many ways, the bacteria share a common ancestor, so all have a few features that are going to be in common. All bacteria are going to be single-celled organisms with no nucleus or organelles, one or more circular molecules of DNA as their genetic material, and they're going to have several methods of exchanging genetic information. Because they're asexual, the biological species concept cannot be applied to them. Thus, bacteria are going to be classified on the basis of physical appearance or, preferably, genetic sequence. So while bacteria are going to be responsible for many diseases like strep throat, cholera, syphilis, pneumonia, botulism, anthrax, leprosy, tuberculosis, disease-causing bacteria are only a small fraction of the domain. And bacteria seem to get less credit for their many positive effects in our lives. Consider E. coli, for example. They're going to live in your gut, and they help your body digest food that you eat. In response or in that process, they're going to make certain vitamins for your body that they need, for example, vitamin K. Other bacteria are going to produce antibiotics such as streptomycin, and so they help to keep you healthy and well. Other bacteria are going to live in symbiotically relationships with things like plants as small fertilizer factories. They can convert nitrogen into a form that is usable by the plant. Bacteria also are going to give taste to many of our foods, things like sour cream and cheese, yogurt, sourdough bread. So increasingly, bacteria are going to be used in biotechnology, from those that can metabolize crude oil and help clean up oil spills to transgenic bacteria that are going to be used to produce things that we need like insulin and other medications. So the archaea domain are going to include many species that live in extreme environments. Where once it was going to be assumed that life could not survive, the archaeans not only existed, but they thrive and they diversify. Analysis of genetic sequence is going to indicate that archaeans and bacteria diverged about 3 billion years ago. Although it's likely that some genetic exchange continued to occur between them, they have evolved in largely independent paths ever since. So approximately 2.5 billion years ago, the eukarya split off and formed the archaea. The archaea are still going to be grouped together, all in one kingdom within the domain of archaea, but we have no idea how many species actually exist. So given that archaea are the dominant microbe in the deep seas, it may very well be that these organisms of which we were completely ignorant of are going to be the most common organisms on Earth. That's still too early to tell because we can't go to the deepest depths of the ocean. So the archaea are going to exhibit some tremendous diversity here. And with this, they're going to be divided into 5 groups that are going to be based on physical features. First, we have the thermophiles. They are the heat lovers. These are going to live in very hot places. Next, we're going to have what's called the halophiles. These are salt lovers. These are going to live in very salty places. Next, we have high and low pH tolerant archaea, and so they like areas that are going to be super acidic or super basic. We have the high pressure tolerant archaea. These are going to be found at depths of about 4,000 meters or 2.5 miles below the ocean surface. That's where the pressure is almost 6,000 pounds per square inch, compared with the air pressure of less than 15 pounds per square inch at sea level. And then we have our methanogens. These are going to be an anaerobic archaea, and they're going to produce methanes. The eukarya domain is going to consist of 4 kingdoms. We're going to have plants, animals, fungi, and protists. There are 3 kingdoms of eukarya that can be seen with the naked eye. We have plants, animals, and fungi. All are going to be made from eukaryotic cells that they have a membrane-enclosed nucleus, and each kingdom is almost entirely multicellular. A 4th kingdom is going to contain the protists, which are often too small to be seen with the naked eye. And it's going to be sort of a grab bag that's going to include a wide range of mostly single-celled eukaryotic organisms. They include things like amoebas, paramecia, and algae. As we noted earlier, biologists now know that protists are not monophyletic groups, and increasingly they're starting to split them into multiple kingdoms within the eukarya. Of about 1.5 million named species, the majority of eukarya, with about half of it being insects, is going to be more result in an interest of bias in biologists than a reflection of relative numbers of actual species in the world. So when you look at that, you're actually going to have more insects than any other animal. So this is going to finish us up for our second part of our video for Unit 1 for Biology 102. I look forward to seeing you back with our next unit.